So today I'm going to talk to you about the regulation of color additives in foods. First, I'll start with the definition of a color additive, and I'll talk about FDA's color additive requirements. And then I'll talk about the color additives used in foods, both synthetic and natural colors, and the trend that we're seeing that we're the use of the natural food colors and some of the challenges. And then I'll end with a quick overview of global regulation. The FDA defines a color additive as a substance that imparts color to food, drug, cosmetic, medical device, or to the human body. We do not consider any color additives to be natural ingredients in foods. That means every color additive is artificially added, and food ingredients that contribute their own colors are not color additives, such as strawberry and strawberry ice cream. So there's many different purposes of food coloring. First, it provides the identity to foods. Maraschino cherries are naturally beige, but red food coloring is added because consumers expect. It also denotes the flavors of candies. For example, green candies denote a lime or apple flavor. They're also for the effect, the bright colors in children's cereals and cake decorations. They're used to mask the natural variation in color. Oranges are normally mottled like orange, brown, and green, but citrus red number 2 is sprayed on the outside of the orange skin to give you that bright orange. It's used for enhancing naturally occurring colors. Astaxanthin is added in salmon feed to give you like a peachy color for the fish, and copper salt is illegally added in olives to give you that bright green color. So color additives are used to protect the products from damage or color loss for such things like light, air, moisture, temperature extremes, and other storage conditions. FDA has regulatory responsibilities for foods, drugs, cosmetics, and medical devices marketed in the US. FDA first must pre-approve the color additives used in these FDA-regulated products. All the permitted colors are listed in the Code of Federal Regulations based on their approved uses, and some color additives are approved for more than one use. So how does a color get FDA approval? Well, let's say a firm wants to have this new color additive. Well, they will submit a color additive petition to FDA, and that firm must provide information on the chemical description and uses, such things as is that color additive made out of. Give us an exposure estimate of the intended uses. They have to give us toxicological studies to show that it's safe, and they do an environmental assessment. Qualified FDA scientific experts review the data, and if the color additive is shown to be safe, it is listed in the Code of Federal Regulations with specifications, uses, and restrictions. And all those specifications come from the petition process. The listed color additives are then in Part 21 CFR, Part 73, 74, and 80, where you can find all the color additives permitted in FDA regulated products. So these are two types. You have your certified colors, which are in Part 74 and 82, and then you have your certification exempt color additives, which are in Part 73, which is your natural sources. So I'm going to go through in detail to discuss each. Certified color additives, these are your synthetic organic ones that are manufactured. And they are denoted with the FD&C nomenclature, which is food, drug, and cosmetics. All of these must conform to the identity and purity requirements that we listed in the CFR based on the petition that was sent. And there's strict limitations to heavy metals, and there's carcinogenic impurities also based on the manufacture of those color. So what happens is these synthetic colors go through FDA's color certification program. It is FDA's oldest user fee program, and it ensures conformity to the purity requirements that are listed in the Code of Federal Regulations for these colors. The manufacturer submits a sample for each new batch. So basically, they send it into the FDA, and the color certification branch conducts the analysis that are listed in the Code of Federal Regulation for that color. And if all the requirements are met, all the specifications are met, that color gets a certificate and a lot number. And we do this all in 5 days. So let's say a red color comes in with the common name of Valora Red AC. It meets all the specifications. It leaves being called FD&C Red number 40. So here's an example. On the right, you can see the manufacturer, the structure of Red 40. And all these specifications are based on the manufacturer of that. So each color is different. And all these are tested in-house. So you can see there's a lot of testing being done. So now let's switch gears to your certification exempt color additives. These are the ones that are generally derived from plant, animal, or mineral sources. And some of these, though, might need purification by processing. These also must conform to identity and purity requirements, but they're not as stringent. There's some strict limitations to heavy metals, and depending on how it's made, there can be some residual solvent specifications or microbial specifications. Again, these are all put in during the petition process. So these are exempt from FDA certification. That means FDA does not test these. It is up to the manufacturer to comply with the specifications. So there's many different types of pigment classes of these natural color additives, from carotenoids in your orange vegetables, like your carrots and your peppers, to your purple vegetables, the anthocyanins in there. And you have a phycocyanine and a cochineal carmine. And these are very different than your synthetic color additives. Here are some other examples. You have your annatto extract. It is made from a red seed, and what happens is seed from this tree is extracted with approved solvents. So then you would expect a residual solvent specification. Another example is a cochineal extract of carmine. It is a red bug, an insect source. And that color is obtained after removing alcohol from the of that female insect. Again, this must be treated to destroy the salmonella. We also have spirulina extract, which is cyanobacterium, and this must be tested for a microcystin toxin. So as you can see, they're all very different. You have inorganic compounds. These are your synthetic iron oxides and your titanium dioxides. And these need to be purified by processing because they may contain, and they also may contain high levels of arsenic. You have your spices, your turmeric, your paprika, and your saffron. Again, these are extracted with approved solvents, so those residual solvents must be tested. And then food crops, which are your fruit and vegetable juices. Now, these can only be extracted with water only, but some manufacturers may be using other solvents, which would not be permitted. Tomato lycopene, however, can be extracted with ethyl acetate, but to a certain limit. So the ideal properties of food colors is that they should be non-toxic and have no physiological activity. There should be a high coloring power, so only small amounts are needed. They should be stable, including light sensitivity, temperature sensitivity, and pH stability. They should be free from taste or odor. Now, these are all characteristics of synthetic food colors. However, consumers want food colors to be more natural. There was a 2007 Southampton study of synthetic food colors. And in this, they looked at children's consumption and possible adverse behavioral effects. Even though FDA and other international authorities have continued to conclude that synthetic food colors are safe for their intended uses, we have seen an increase in a trend towards using colors from natural sources. There's a 36% increase in Europe and a 28% in the US. But there's challenges in using these colors from natural. Products should look and taste the same as products with synthetic food colors, and that doesn't always happen. If you look at the bottom here, the bottom right is you're using synthetic colors, and your bottom left of the cereal is using natural colors. You can't get the blues that you normally would. Some of these colors have have shelf life issues, and in time, the color may fade. They have less coloring power. So a relatively larger amount is needed to achieve the desired color, which may affect the taste depending on the source of that color additive. Some are less stable and more variable in shade than synthetic color additives, and they can vary in composition from batch to batch. Also, fruits and vegetables are food crops, and there's a likelihood that pesticide residues are present. Some of these pesticides may be banned in the US. And when we're making these juices, usually they're extracted from food crops and concentrated. And that means there's a potential for a higher amount. Solvents other than water may be needed to express the juices. Therefore, the juices may have non-permitted solvent residues. So quickly, I'm going to go over how color additives are regulated globally. Now, overall, the process is similar. We looked at 7 different countries, and they're all based on the assessment of safety for the intended uses. However, there's different regulatory frameworks. The responsibility for rulemaking is different. How they define a food color, the approval process is different. What is needed for approval is different. And therefore, the end result is different. There's different food colors and specifications and different restrictions for use. Here you see a table of some permitted food colors over these 7 different countries. And as you can see, they're not consistent across the board. They are permitted in some countries, but not others. And more specifically, here is an example of that. This is the curcumin and a turmeric regulation. So in the US, the color additive is called turmeric, and it's allowed in foods generally at a GMP level, and they're specific. However, in the EU, it's called curcumin, and it's allowed in some specific foods, different levels, and the specifications are different than the US. So as you can see here, the regulations are different across the different countries. And with that, I will end, but we can have questions at the end of this session. Thank you.
Hello, today I'm going to talk about the industry perspective on the growing concerns and a trend of using colors from natural sources. My name is Mark Goldschmidt. I'm Quality and Product Safety for Sensient Technologies. And as you can see on the screen here, there's several hazards or risk factors that come into play with natural colors as opposed to the colors that are certified by the FDA. They're microbiology, heavy metals, pesticide residues, residual solvents, or adulterations. So I'm going to talk about a few of these risk factors. So I'll go ahead and turn on the various countries and what the percentage are of consumers that report to be are extremely concerned about food colorings. So you can see, some countries, in particular, China and Russia, I was rather surprised to see the numbers that high. But there's always concerns about the types of colors that are being used, and in fact, that's Botke's presentation on the history of why colors are certified is for this very reason. The colors that undergo certification. And obviously, these colors are not required to undergo FDA certification. So issues facing the color industry is the Southampton study, which many can argue about the validity of that study, but it certainly, the consumers are going to drive the use of natural colors or colors from natural sources. And the trend seems to be growing in that direction, as we'll see here shortly. Supply, environmental concerns about synthetic raw materials, basically sustainability of the renewable natural colors, and then ongoing food safety issues, which I'll cover here in a little bit. And then the lack of specific quality and safety specifications in the colors exempt from certification, or as I'm referring to, colors from natural sources. Climate change could be a factor affecting quality and availability. And then I'll show you a sampling study of products represented as colors in the marketplace some years ago. And then a slide on import detentions of food products that involve color. So here is, you'll see that the regions of the world that are amenable to crop, should say good quality crops, is green. So you can see there's probably about 70–75% of the world either too cold or too hot to really grow these types of colors or crops that we can extract color from. And with climate change, that trend we believe is growing as far as regions of the world that will be able to grow these. So food safety issues in the news, contamination issues, and I have both contamination and adulteration. The difference being the adulteration is intentional, where contamination in most instances is unintentional. So extraction solvents approved for colors that can vary by region of the world. Excess residual solvents, sometimes in excess of 1,000 ppm. Lead and mercury contamination in carmine, and pesticide residues in paprika, dioxin in paprika, and bacteria pathogen contamination. And then adult issues that seen in the marketplace over the years is beverage cloud adulteration with plasticizer, and that was for shelf life and cost. Synthetic dye adulteration with natural colors, Carthamus adulteration with orange 2, saffron, a very expensive natural color adulterated with Carthamus, and brick, yes, brick from your house in paprika, and in Sudan dyes and turmeric and paprika. And that goes back to 2005. It's probably happened 2 or 3 times since then. It kind of happens, goes away, and it happens again. Sudan dyes are carcinogenic textile dyes. So here's a study that was done in India. Basically, they had sampled about 74,000 different samples, and about 1 in 5 was found adulterated or misbranded. And this, some of the sampling actually happens in the U.S. I know the New York State Lab does a lot of this. They'll go out to these ethnic markets and sample stuff that looks like it has color in it, send it back to their lab, and they see similar issues. So this was a study that Sensient did back in some years ago when we were looking at a dual sourcing initiative. So basically supply security. So we sampled different products in the marketplace that were represented as colors, color raw materials, fruit extract, vegetable extract, spices, and carmine. And you can see the issues that we discovered here. And this was about 1 in 5 that was considered to be adulterated or contaminated. Micro is in gray, adulteration in purple, heavy metals in green, pesticide residues in yellow, and residual solvents in red. So you can see the issue by color grouping that we observed. And this was a rather interesting discussion. We would go back to these people and say, hey, we found this issue. And they say, well, okay, if you don't want it, we'll just sell it to so-and-so, because they don't check that or they don't care about that. Hence the reason for these standards on colors from natural sources. So this is detention of incoming food products due to color. It continues to grow. The navy blue bar is undeclared color, our labeling issue, and the yellow bar is illegal dyes. So you can see that trend continues to grow. So as I mentioned, the need for colors from natural sourcing, having some agreed upon standards within the color industry, and should be enforced by customers of the color. Since we got this feedback when we did this dual sourcing initiative, we embraced a group of individuals that would be considered experts on this topic. So you can see the committee, an expert committee was brought together to look at some proposed standards on colors from natural sources, and that's the committee there. And the link to the white paper that was published out of this committee is there below, and here is the abstract. So this committee came together and said, yep, what you're doing here is exactly what should be done in the marketplace. And some of these individuals, most notably Jim Simon, Mario Ferruzzi, Steve Talcott, and Monica Giusti, were all experts in either color are also dietary supplement standards, which they believe this should take a similar approach. So when you look at using colors to make a product that we sell to the consumer products industry, there are several risk factors that you need to keep in mind from a microbiological perspective, just to make sure the risk is being controlled. What is the customer's application or specification? Is the material a synthetic or from natural sources? This is obviously from natural sources, so that's a risk factor that's in play. Microbial control step, and this is FSMA Preventive Controls Rule, which is an established rule for some years now, so it's law. Nutrient content, if there's nutrient content in the color or product, it will sustain growth. pH control, if you have a low pH, that can control the risk. Typically in the neutral range is a good growth region for micro. Water activity, the lower the better. Salt content, higher salt functions as a preservative. And then is the product preservative additive or preservative free? If it's preservative free, then that's higher risk. Storage conditions, which include just packaging and whether it's refrigerated, frozen, can control the risk. And then there are various resources within the USDA, FDA, and European regulatory bodies that you can look to see if there's been issues with this particular type of product. So this is an example of a risk matrix. So you can see the various categories I discussed on the previous slide: natural versus botanical or synthetic. Import alerts, has there been an issue in the past? Kill step, preventive control, contains a preservative, yes, no, contains nutrients, basically what level of nutrient is in this type of product. Water activity, if it's high, then it's higher risk, and in pH, if it's in the neutral range, then it's a higher risk. So anywhere where you see text in red, that means that particular factor is high risk. So what you see below is the risk level for each commodity. So turmeric, high, paprika, high, fruit and vegetable juice is high, and annatto is high. So below that, you see the recommended testing that you would do on each of those types of colors. So identification, heavy metals, microbiological, pesticide residues, residual solvents, and adulteration. All these products have, in the past, have seen some issue with adulteration. So managing critical factors in the natural colors, microbiological, these come from crop-based products. So you want to control the pathogen potential. Heavy metals, again, comes from a crop, so you can have heavy metals in contamination in the soil. So you want to screen for that. Same way for pesticides. That's compliance with the EU and EPA. Adulteration, test for synthetic dyes, and there's most of the labs have a illegal dye screen that they run nowadays. So you want to screen for those types of known adulterants. Also, there are types of testing that you can do to just look for something different and then flag it. Unauthorized solvents, again, making sure the solvents that were used to extract it are what is listed in the regulation. Supplier reliability and raw material traceability. So I believe that is my last slide. So I'll wrap up from there. Thank you for listening and tuning in. Thank you.
I'm Joe Konchnik with ResTech, and I'm going to talk about the challenges with developing methods for color additives from natural sources. If there's one word I had to use to describe what it's like to develop methods for these types of materials, I would use the word complexity. And we'll take a look at 5 different supporting reasons why I chose that word. First, I'll talk about the variety of the materials of sources that tend to be used for the manufacture of these materials. Next, we'll talk about the matrix characteristics that are specific to each and how they impact method development. Then talk a little bit about selecting the right analytes and what a challenge that can be when we don't have regulations for these materials. Achieving limits of detection, I'll cover a little bit about how we can take existing methods and apply them to these materials and see completely different results. And then finally finish up with a gap in the industry currently, which is a lack of reference materials. So let's begin with looking at the variety of these materials and their sources. As Bhakti mentioned earlier, there's a great variety of these materials from different types of plants, and you'll notice even on this slide at the bottom, carmine is derived from insects. But for the most part, most of our research to date has been developing methods for plant-based natural color additives. So you can see from what I have pictured here, when we're talking about plants, the colors can be derived either from the plant leaves, from seeds, from the fruits of the plants, from the roots or rhizomes, and also from vegetables. So this great variety adds complexity to developing methods, because one of the first things we try to do as chemists when developing methods is narrowing the scope and trying to simplify the complex. For these materials, this is quite a challenge. Let's take a look now at some of these matrix characteristics and why they're a problem. In comparison, if you look at the FDA certified synthetic colors and look at the methods for analyzing these, you'll note that these are very well-defined materials. They're manufactured. We know what's in them for the most part. We know how much of each chemical is in these materials. And the methods are very well described. And because they're manufactured under very tightly controlled manufacturing processes, we pretty much know what to expect. In comparison, when we're working with these natural color additive materials, we have a variety of additives, which are used for many different things. We have a variety of manufacturing processes and sources from which these materials come. So a couple of examples, if you look at annatto extract, it's a solvent extraction that comes from annatto seed. Then maybe take a look at carrot oil, which is a hexane extraction from carrots. And then contrast that perhaps with turmeric, which is a ground rhizome powder. So, and then there's various other modifications of these as well, other solvent extractions and oleoresins. And you can see that in these photos below, you can see that there's quite a variety between ground powders, oils, and solvent extracts that need to be tested. And this adds complexity for the following reasons. Some of the matrix challenges we'll see involve working with dried powders. If you've ever tried to weigh a dried powder, it can be a little difficult working with these materials. And then extracting them. Adding solvent or wetting them can be a challenge because of the nature of these materials and the physics around these particles. So one has to be careful in properly wetting them. Also, obtaining a representative sample can be a challenge. Making sure the sample is homogeneous and is properly mixed can be quite a challenge when you have so many different types of materials to work with. Also, fine particles may clog analytical systems and leave residues. This can contribute to system shutdowns. It can require maintenance and maybe changing some of the consumables to keep the method running. It also might mean carryover from one sample to the next. Oleoresins contain solvents and often sometimes oils, so we have to be careful that these materials are matched properly with the solvents normally used for workflows of these types, and that can be another challenge. Knowing that these are concentrates adds another dimension of complexity, because what's concentrated are the plant materials themselves, in addition to the contaminants that are present. Having higher levels of plant matrix present or higher levels of contaminants affects the dynamic range of calibration and can affect limits of quantitation. MS detectors are known for being used quite readily and extensively within food testing. And it's no exception here for chemical testing. And something we have to be concerned about there is ion enhancement and ion suppression, which can be caused by excess matrix in the samples. So this can also lead to poor accuracy and precision, and also due to the nature of handling each matrix, having to handle these materials because they're so different manually might affect the precision, whereas normally for other types of materials that are very well defined, they can be batched in an automated process, but that may not be the case with these materials. Next, let's take a look at some of the challenges we've had with selecting the right analyte list for some of our work. Within the AOAC colors community, we've started working in 4 subgroups for microbial contaminants, pesticide residues, residual solvents, and heavy metals. We'll take a look at each of these. First, for the microbial contaminant subgroup, note that there are quite a few different pathogens that need to be looked at, different organisms. And each of these have to be handled a little bit differently. So you can't develop one method to test for all of them. This subgroup has been working on this for probably the better part of the past year and has completed their salmonella method, but they still have to work on listeria, E. coli, Staph aureus, yeasts and molds, etc. So that means sometimes you're not just going to be developing one method, you're going to have to develop multiple methods in order to cover all the contaminant groups and possibly all the different sample types. For pesticides, we were met with an initial challenge in our subgroup, because that's one of the ones I'm involved with, where we started off with trying to pare down the list of pesticides one could analyze for in these materials. Very typically, labs out there in the community analyze upwards of 500 or even 1,000 different pesticides, depending on commodities. So we launched into a procedure where we had, through a collaborative effort, people shared lists from various state, federal agencies, some of the different countries throughout Europe and Asia. We looked at the FDA's list, and we also looked at some lists from manufacturers and private testing labs, put them side by side, compared them to what types of reference materials were available out in the community that we could use for spiking studies. And we came up with over 650 pesticides total. So then there was a sort performed, and we looked at which of these compounds appeared mostly across all these lists. And we were able to decrease the list from 650 down to 72 of the highest priority analytes. So this, you could imagine, wasn't an easy task. It took quite a bit of coordination and collaboration and time, but it is a step that has to be taken when working with materials like these that don't necessarily have regulations set for them. Likewise, in the residual solvents subgroup, to a lesser extent, they started with the USP 467 list, which is a method that's typically applied throughout the industry for analysis of pharmaceutical raw materials and ingredients, and contains a list of 36 solvents of various classes. And they were able to narrow it down to 12 authorized solvents, looking at which solvents appeared most frequently across the different regulations for each of the color types. So through this exercise, we were able to see that looks like anywhere between 25 and 50 in most cases would be the regulatory levels that this group would have to focus on. Again, this was time consuming and took some time to sort out. Likewise, in the heavy metal subgroup, which is one of our newest subgroups within the community, they started looking at which metals were showing up mostly in different regulations between the US and Europe, and then matching those with what the regulations say for each of the color types. And again, going through this process of narrowing the analyte list can be a challenging, can be time consuming in the absence of regulations. Let's take now a look at achieving limits of detection. One example I chose for this was the residual solvents subgroup, where we had one of the laboratories volunteer to participate in a proof of concept study, taking that USP 467 residual solvents method, modifying it for these materials. And you'll note that they decided to bracket a range between that 30 to 50 ppm range. They bracketed from 10 to 100 ppm and spiked at those different levels, these solvents. And note that they didn't just use the approved solvents, they also used some solvents that aren't approved for these materials because we thought we should be looking for those as well. We looked at challenging the method with one control, which is the soybean oil, and 5 color samples. And you can see that a method that works quite well on pharmaceutical ingredients doesn't work as well when you challenge them with these different types of powders, oleoresins, and solvent-based materials. So it does tell us that there are quite a few challenges ahead as we continue to work on this method. And finally, the lack of reference materials in the community is a glaring gap. Reference materials are used quite often to verify and validate methods and even cross-validate methods among multiple laboratories before their methods are passed as final approved methods. And so we take a look at some of the SRMs, standard reference materials that NIST produces. They have some for rhizomes and such as ginger and turmeric, for some metals, and also one for trace elements and spinach leaves. And then we take a look at the FAPAS program in the UK, has one reference material for 3 metals and turmeric, which are useful, but don't necessarily, may not cover the entire scope of what it is we're trying to test for. So there's a gap in the need there. And then as I went out and searched for SRMs for solvents, pesticides, and microorganisms, I found nothing. So this is going to be a continuing need in the future to have these reference materials identified, developed, and made available to the community to verify and validate methods. So in summary, the complexity of developing methods for colors is may be supported by knowing that there's a very wide variety of these materials and their composition varies greatly. There's a lack of regulated analytes, a lack of regulatory action levels for these, so it makes it difficult to develop methods when we're sort of having to base it on other things. And then there's a lack of reference materials that needs to be addressed in the community. And finally, we believe that collaboration will drive innovation to get these methods developed. And we believe that community engagement will drive the solutions. So I want to thank you for listening and look forward to answering your questions.
Hi, and I'm John Kloss with the Coca-Cola Company. And I will be finishing up this webinar with the benefits of the color community to food and manufacturers. So the purpose of the color community is to support the AOAC mission to promote method development and validation. And we're doing this by establishing an international network of analytical professionals who share a common interest in development and validation of color additive methods. So the stakeholders include state and local governments and federal governments, standard setting agencies, and industry reps. So the benefits of establishing these standards are higher quality color sources available to processors, developing and harmonizing standards that will stimulate trade, ensuring consumer product safety, better definition of product quality, guidance for sourcing, brand protection of all companies in the supply chain, and availability and reference by all in the industry. So by doing this, will stimulate industry self-policing, thus preventing further enforcement or restricting trade in natural colors. So how can this community help? By developing the needed methods or extending existing methods for color additives, providing color additive samples, participate in round robin validation studies, and maybe most importantly, prioritizing efforts of the community. So you can see on the right, the group of companies represented on this committee, and they include color additive manufacturers, instrument and consumable suppliers, food manufacturers, contract laboratories, FDA as well as USP, FCC, and they all have an emphasis on color additives from natural sources used in foods. So the areas of interest of the color community are microbial contaminants, pesticides, residual solvents, heavy metals, and adulteration with synthetic dyes and other materials. So as for microbial contaminants, the areas are Salmonella, Listeria, E. coli, Staphylococcus, yeast and mold, and total plant count as a quality indicator. So for Salmonella species, to give you an update on the progress for it, a method for screening has been developed using the gene-up technology, and 10 color additives have been tested. And all the products demonstrated acceptable recovery and detection of target organisms. So the next step is to add turmeric to the existing method through matrix extension. And an oral presentation was given last year at the AOAC virtual conference by BioMaru. Excuse me. For residue analysis, a stakeholder working group was created of government agencies, natural color manufacturers, contract labs, and instrument and consumable suppliers. And the list of pesticides was narrowed as well as identifying color samples for method development. So this gives you a timeline for the work that's been done in this area of residue. So about a year ago, a draft SMPR was created to describe method development activities. In addition, manufacturers donated samples and shipped them to volunteer labs, and consumable suppliers also donated calibration and spiking materials. And then last summer, proof of concept testing was completed by 2 volunteer labs. And an oral presentation on this was also given last year at the AOAC conference by Matrix Pacific Ag Laboratories. So for residual solvents, to give you an update, static headspace GCMS method has been developed for the 14 solvents, 11 of which are authorized and 3 are not. So 8 natural color products were tested and using DMA water solvent system with chlorobenzene as the internal standard. And a presentation was also given at AOAC last year by ALS Truesdale Labs. And a SMPR is being drafted for this as well. In addition, there was a poster given at the AOAC conference in 2019 on this subject. So for heavy metals, to update you, a stakeholder working group was also created for this as well, and the list of analytes was worked on for the colors. The plan is to extend the current method for analysis of lead in turmeric by ICP-MS to additional metals and colors. And for this, also manufacturers ship natural color samples to volunteer labs. And finally, we're evaluating several other methods for other colors, additives, as well as other heavy metals. And an SMPR is also being drafted for this. So for adulteration with synthetic dyes, since we are talking about crop-based color additives, they are subject to variation and supply issues due to growing region and climate. Therefore, they are prime targets for adulteration with synthetic dyes. So currently, there are tests available that are targeted analyses, but non-targeted methods are needed. So the AOAC authenticity fraud groups are working on this regarding spices. So FCC does have some new non-targeted screening methods, one of which is by TLC, and this provides a quick and inexpensive detection of synthetic dyes illegally added to spices in the 100 ppm levels. In addition, FCC has an LCMS method that provides accurate detection at ppm levels of illegally added synthetic dyes. So this kind of summarizes what I've been talking about, and I obviously won't go through all of it, but it shows for the solvents and the color, or I'm sorry, the solvents as well as the other areas of interest, what we've done so far. And you can see that quite a bit of work has been done, but there is still quite a bit of work that's needed. So why do we need these methods? So we've talked a little bit about this already, but to give you a visual example, say, you know, we have regulators, the food manufacturers, as well as suppliers. Without any agreed upon methods, you end up with a mix of everyone using their own methods and maybe having multiple methods for different suppliers, different food manufacturers, and so forth. And what this leads to is a reduced efficiency of the labs. Data is difficult to compare between methods or between companies. This is obviously more costly, but maybe most importantly, you end up with spotty coverage of specifications where for various reasons, people might not look at the same needs or impurities or levels, or it just doesn't apply to them. So, however, if we do have universally agreed upon methods from trusted organizations such as the AOAC, everyone ends up using these same methods. So it increases the efficiency of the labs, and it makes it easier to compare the data between labs. And of course, this leads to reduced costs, but probably most importantly, you end up with better specifications overall for the colors. So why should you get involved? So we have input from many suppliers, but few manufacturers, and the ones that do, obviously, for instance, I'm with the Coca-Cola company, so we would be coming at it from the view of our needs and not necessarily other packaged goods or other types of food applications. So we don't just need your help. We need your knowledge, your experience, and your perspective on these. So we'd really like for you to get involved. So again, want to thank these contributors for their volunteer work on this important community. And if you would like to get involved, please contact one of us and we will be happy to help you get involved. Thank you.